Right. Hi, everyone. For those of you who've joined already, we'll get started in 1 minute. All right. Just going to wait another 30 seconds or so for folks to join. We'll get started.
Hey, Elena, can you take a peek at the chat?
Yeah. Okay. All right. Here we go. So welcome, everyone, and thank you all for joining today. My name is Helena McMonigal, and I'm one of the organizers of this year's Quantitative Seminar series. So I'll begin by acknowledging the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. It is my pleasure to introduce our first winter quantitative seminar speaker today, Tom Langbehn. Tom is a postdoctoral researcher at the University of Bergen in Norway. He studies evolutionary ecology and global change biology, such as how climate change and human activities shape biological processes. Tom combines mechanistic modeling informed by theory with field observation, and he is particularly interested in studying polar ecosystems and the ocean's twilight zone or mesopelagic zone. Today, we'll hear Tom speak about his use of mechanistic models to predict how climate change will drive shifting species distributions and how light may drive range shifts in quite a different way than rising temperatures. A note for the audience, Tom is happy to take questions as we go, so please feel free to post your questions using the chat box. And if any questions aren't covered during the talk, we'll, one of us will reread them as time allows at the end of the talk. Or please feel free to just, post your questions at the end and we'll field questions. So I've had the pleasure of working with Tom in the past and have been fascinated by his research and methods. So Tom, we look forward to your talk and thanks for joining us all the way from Norway, especially since it's 9:30 at night your time. So with that, I'll hand it over to you.
Thank you very much, Elena. It's a pleasure to be here tonight for you daytime. It's good to see that so many of you could join. I have already seen also quite a few familiar names in the list. And yeah, I'm very happy that I get the chance to present a bit of our recent work here today. If I say we, there's a whole bunch of other brains behind what I'm going to talk about. Those are my fantastic collaborators down here that I want to acknowledge. So this is very much a joint effort. And yeah, for me, these kind of seminars have been kind of motivation and inspiration during the last year with lots of home office. So I'm very glad that I hopefully will be able to give back a bit of inspiration today. What I want to talk about, I have grouped in 3 sections today. I will start with talking a bit about the objectives for this talk and introducing some of the methodology. And I will use some more time about this than I usually spend on it, very much in the spirit of this quantitative seminar series. And then I will use 2 case scenarios. I will focus a bit on the first one, and if the time allows, we will look at the second one, which makes use of these mechanistic models to draw some conclusions about species redistributions. And I will try to, at least after each of these parts, have a look at the chat window. So I have 2 screens, so I might not see immediately if a question there in the chat pops up, but I will try to have a look every now and then in between, and then hopefully pause and be able to answer those questions. Yes, species globally are on the move. This is probably one of the most ubiquitous responses to climate change. We see this happening both in the ocean and on land, and generally those range shifts are directed polewards in the oceans or towards greater depth. And what this means is that species are being observed to track climate velocities across latitudinal gradients. So a species that tracks a climate velocity tries to maintain a constant temperature. And in this case, for example, here species A, to maintain an optimal temperature at 270 degrees would, under a warming scenario, have to shift northwards about 10 degrees to maintain that same temperature window. At the same time, a species at higher latitudes would need to shift much less in terms of distance to maintain the same temperature window. The models that are mostly being used for this kind of species distribution modeling are based on statistical correlation, assuming a fixed relationship between a temperature niche and the distribution of a species. And there are many other factors then, of course, that are neglected. And one such factor that changes drastically across latitudes is, for example, light. And I hope I will be able to convince you that this is an important factor to consider, especially at the higher latitudes. So as you can see, what I've plotted here in blue is the change in day lengths. At the equator, days are equally long, around 12 hours. But then as we move towards the poles, we see that the day length diverges seasonally. And once we cross the Arctic Circle, we will get days that exceed the 24 hours. So this is important because it means that the species closer to the equator, while shifting a longer distance, will experience relatively little change in light regime, while a species closer to the pole, although shifting much shorter latitudinal distance, will experience a much larger change in the light regime. And it's actually not only the 24-hour light cycle that matters here, because these seasons, of course, extend. So already 10 degrees above the Arctic Circle, the photoperiod will extend to 3 months, 3 months of midnight sun during the summertime, and conversely then also 3 months of polar night during the wintertime. So these are environments that are very different from the circadian light cycle at lower latitudes, and they require different physiological, morphological, but also different life history adaptations that are generally not considered in these statistical type of models. There have been, however, some recent work that actually highlight those points and have posed the idea of photoperiod constraints to species-driven range shifts, but to our knowledge, nobody has actually quantified or tried to model these photic barriers. Light is an important factor to consider in terms of seasonality, because it has a twofold impact on life in the ocean. It acts at the very bottom of the food web, providing the energetic basis for all higher trophic levels, but it also has a top-down controlling effect, because more light will yield better vision and therefore better foraging opportunities. And visual forages can be found all along the marine food chain. We have visual forages down at the invertebrate level, amphipods and krill, for example, and then many top predators from seabirds, fish. Here we find, for example, that some of the larger species even have developed cranial endothermy for keeping their eyes warm, so it's a very important part for them. But also some of the baleen whales are even reported to use vision, at least contrast-based, to locate swarms of krill. And it is these interactions that we try to quantify, so we can try to model prey encounter. And I believe that if we want to move from correlation to causation, then we need to study the mechanism. And sensing, in this case, visual sensing, is a very important part, because without sensing, there is eventually no energy that is acquired that can then be distributed to the different functions through routine metabolism, but also to structural growth or reproduction. So sensing is really key to make energy available to the organisms. And this is exactly what we are trying to do in the type of models that we are using. We are trying to integrate physiology with behavioral and ecology, but also species interactions. In the model that I have developed during my PhD, we consider 3 trophic levels. We have a piscivore, which in our model is parameterized for a cod-like predator. The tuna silhouette is just a bit more recognizable here. We have a planktivore, which in the model is parameterized as a small mesopelagic fish. It's the glacial lanternfish, Benthosema glaciale, which is one of the most abundant mesopelagic species in the North Atlantic. The 3rd trophic level is zooplankton, which we here consider to be of the genus Calanus. And for these trophic levels, we model the encounter rates, and these ones are dependent on the light regime. Light enters the equation here as it scales the visual range of the organisms, which is also dependent on the contrast and the area of the prey and the eye sensitivity of the predator. These visual models, I could fill an entire seminar with them, but Sean Rowan, one of the organizers, has actually done this already. I think it was last year, and the recordings are still available here in the series. So I'm going to skip over the visual models, and I recommend everybody to take a look and rewatch that recording of his seminar. What I also want to point out is that the encounter rate between planktivore and zooplankton in our case is also handling time constrained. So there's a density dependence. At higher prey concentrations, the encounter rates become satiated because the planktivores are no longer able to handle all the prey that they will be encountering. We then continue to model the gut dynamics of our focal species of the planktivore here. So prey can only be encountered if the stomach is also evacuated. And in this case, this gastric evacuation rate is a function of temperature. We use a function that is actually species specific in this case for myptophids, for this type of lanternfish that we are looking at. And the acquired energy is then distributed in a 2-step process, first towards the basal metabolism and to fuel routine behaviors. And if those energetic demands are met, then in the model we allocate the acquired energy to reserves. These reserves act as a buffer and can be drawn from if the metabolic rate exceeds the acquired energy. And again, this metabolic rate that we are using here is both dependent on weight, but also has a temperature dependent term in there, and scales with a Q10 of about 2.5 in our model. If the fish are able to maintain full reserves, the surplus energy can be allocated to growth, reproduction, or migration, for example. And this is a very important term because this is one of the fitness related parameters that we will focus on for the modeling. If the reserves are empty, this will directly affect the survival of the species, which is the second fitness related term that we will be looking at. Survival is also affected through predation, which is a function of the encounter rate with the predators. Both survival and surplus in our case enter as a proxy for fitness here. So we're looking at lifetime reproductive fitness, which is a proxy for Darwinian fitness. And we are maximizing this using a dynamic optimization algorithm. And using this dynamic optimization algorithm, we will see that there are emergent strategies. And I'm going to spend a few more minutes on explaining what dynamic optimization is. Our model is placed in a 1D water column environment, and this environment is vertically structured, but also seasonally. So we have a temperature field, which we get from the World Ocean Atlas database. So it's a climatology. We have a light field, which is resolved in hourly time steps, which we model. And we have a prey field, which is informed by empirical data. Generally, surface waters in our model are warmer. They bear a higher predation risk because there's generally more light, but they also have higher prey encounters because there is more light again, and also because the copepods tend to aggregate in surface waters during summer. With increasing depths, it becomes cooler, it is less risky, but also the prey encounter goes down. So down there, it is a more safe environment, but with less rewards. The model is resolved for the full entire cycle, and we do that in hourly time step over a water column between 0 and 1,000 meter in steps of 10 meters. And what we are asking from the model is to find the optimal strategy to get from time step 1 to the end of the year while staying alive, and also maximize the surplus energy. So we are looking at these two fitness parameters that I have shown in the previous slide. And as you might see, there are a lot of possible combinations here. At every time step, our model organism is allowed to choose a depth freely. The only constraint is that they cannot migrate more than 100 meters per time step in this model. But else they are free to choose at every time step the depth locations that maximizes their fitness. And it's very important that in this case, we are not looking at the immediate reward, but we are looking at the reward at the end of the season. And to find these optimal strategies, we are using the dynamic optimization algorithm that I already mentioned, which I will explain. And we are doing this for in total 10 different gut stage, but also for 10 different reserve stages. Because the decision that an individual with a full stomach will make are different from those that an individual will do that is in more desperate need, for example, to find food. To use dynamic optimizations, or dynamic optimization is useful when individuals make multiple decisions over time and the past decisions are influenced the future. And as I said already before, the optimal act is not the one that maximizes the immediate reward, but it's directed towards the long-term rewards. And the best actions in any time step depend not on how you ended up in a state, but depend not on how you entered the current state, but how you behave forward in time. And to illustrate this and make it a bit more visual, I'm going to use an example from a book by Hannah Coco that I would really recommend for anybody who is interested in taking up this kind of modeling. We will be looking at a PhD student traveling across Europe. For those that are a bit more versed already with modeling, I of course highly recommend the Bible of dynamic optimization modeling, which is the book by Mark Mengel and Colin Clark. We are very lucky in Bergen that Mark is a longtime friend of the group and is affiliated with the group, and he has been an amazing mentor for many of us early career researchers. So I highly recommend that book as well. So for the example, we are interested in how a PhD student travels from Barcelona to Jyväskylä in Finland, and there are multiple options that this student can take. And we can project these travel routes a bit more neatly along the same axis that we were going to be using in the model. So we have a time axis and we have a space axis here. And the question is, what is the optimal route to get to the final destination? And in order to do that, we need to define a common currency. In this case, we score happiness points. In our mesopelagic fish model, it is the trade-off that relates to fitness. These fitness points or these happiness points in this case are scored based on the experience arriving in a city, but also are awarded on how pleasant the way to the city is. So as you can see, for example, arriving from Milan and Hamburg scores very little points. Maybe the student has been in Hamburg before and the travel from Milan to Hamburg is a boring train trip. While, for example, from Paris to Hamburg, he could hitch a ride with a friend that he hasn't met in a long time. So it's nice to reconnect, and that scores a lot of happiness points. Obviously, we could solve this problem forward in time. We could just travel along all these routes, sum up the points, and figure out which is the optimal solution. This is possible in this example because there are just a few combinations here, but once it becomes more complex, once we expand the possibilities, we will end up with millions of combinations. And this is where the dynamic optimization becomes very helpful because it solves the problem backwards in time. So we're going to start at the end, and at every step in time, we have to ask, in this case, if we ever arrive in one of these cities, what is the best way forward from there? And then we add up the points. So for the last time step, it doesn't help us very much because there's only one option forwards from each of these cities. So we're going to go one time step further back in time, that we end up in Stockholm. And we ask again, so what is, if the student ever ends up in Stockholm, what is the best way forward in time? And in this case, there are 4 options. He can travel via Turku, which will give him a total of 2 happiness points. He can take the route via Helsinki, which is not as exciting and only scores 1 point. So from Stockholm, we know that traveling via Helsinki will never be part of the optimal solution. So we can remove actually this line from the graph. So we know if the student ever arrives in Stockholm, the best way forward is going via Turku. We can ask the same question for Berlin. Here the options are to go or to travel via St. Petersburg, which scores a total of 4 points, or via Helsinki, which scores only 1 point. So again, we can remove this route. We take one more example, going to Paris in this case. From Paris, there are multiple options. One scores 14 points. The other one, zigzagging across Europe, scores 13 points. And then there's the option going via Hamburg, Stockholm, Turku, which scores 18 points. So we remove all the options that score less than 18 points in this case. And we can solve this entire travel plan backwards. And then we end up in the end with one optimal strategy, which takes us from Barcelona to Paris, to Hamburg, to Stockholm, to Turku, to Jyväskylä. At this point in time, we would start simulating forward, the strategy forward in time, which is counting up the points in this case. But it's very neat because we have solved the problem backwards. We always retain also an alternative strategy. Should the student in Barcelona get in the wrong train and end up in Milan instead of Paris, we know what the best route forward from Milan would be. It is not to try to get back on the original track, but it is to travel forward from Milan to Berlin, to St. Petersburg, and then to Jyväskylä. And this is the last point here. It does not matter how you end up in the current state, but you should behave optimally forward in time. We are, of course, not only interested how one student would take this trip. We could do the same exercise with a whole cohort of PhD students. They would all have different experience. They are in different states, if you will. So they would score these happiness points differently. And if we then simulate forward in time again, we would see that all these different individual plans would have population effects that emerge from there. And this is the logic that we also apply in our dynamic fish model. Students in this case are fish. We are not traveling across Europe. We are, the time axis is just the full year, and then the space axis in our case is the depth variable. And that brings me to the end of the first section, and I'm taking a look at the questions. So far, no questions. Are there any questions by now? If not, then I will just continue. So we are interested. So we are interested how this model compares to reality. These mechanistic models are, other than statistical models, correlation models, not fitted to data, but we confront them with data. So lucky for us, we had already a published data set that has a high resolution acoustic transect here along the Norwegian Sea, crossing the Arctic Circle, going from the Norwegian coast towards the north of Iceland. On the lower panel here, you see acoustic backscattering, which you can see migrates up and down in the water column as the light regime changes. And we know, because this system is fairly well researched, that the upper of these two scattering layers is most likely composed of Maurolicus milleri, which is a small mesopelagic species. At the lower level is most likely Benthosema glaciale, which is the species that we have parameterized the model for, and some pelagic shrimps. And what the authors, Norheim et al., concluded from their paper, and based on their observation of surface light, is that these deep scattering layers in the Norwegian Sea seem to track a light comfort zone. So they try to maintain always the same light intensities while migrating up and down in the water column. And when you look at the observed surface light, one might notice that we see that the nights are becoming lighter towards the north of the transect here, and also as we're moving further in time, we're getting closer to midsummer, and at these latitudes we see that the nights are becoming lighter. And this was what the authors concluded was most likely driving also this deepening in the mesopelagic scattering layers. However, the interesting ecological question here is that we also see a strong temperature gradient, with the southern end of the transect being around 6 degrees, and then as we move north, we are getting closer to 0 degrees. So it can't be excluded that also temperature is the driving factor here. So what we did is we applied our model to the same data. We actually used the temperature data as input in this case, the light field we modeled, and then we compared the emergent vertical migration strategies to what we see in the data. And as you can see, I've plotted here again with the dashed lines around the light comfort zone as they were described in the paper, and you can see that the model predicts a very good fit to what has been observed via acoustics. The question then is, how can we tease apart light and temperature? Because we can use models as virtual laboratories, we can turn on and off different environmental factors. In this case, we can, for example, simulate how would the behavior change if we keep temperature stable. And we have done this. If we keep temperature stable, not much changes in this model. If we turn off the light, however, and keep the light environment stable without the day-night cycle, we see that all vertical migrations stop in the model. So what actually drives this downwards migration of the scattering layers is the increasing light levels indeed, but there's also a deepening of the daytime values. In this case, this is not driven by the change in the light regime, but it's a change in water clarity, with the north of the transect being in clearer waters than the more coastal waters, which then explains why light penetrates deeper in those areas. It is, however, not only the behavior and the physics that change along this transect, there's also an observed change in biomass. Around the same orders of latitude, here we see that there is a 5 to 6 order magnitude decline in mesopelagic fish biomass. And north of Svalbard, there are studies that report that less than 2 of the total biomass is actually composed by this small mesopelagic fish. And this phenomena is not only found in the Norwegian Sea in our study system, but on both hemispheres. There are several papers that report this decline in mesopelagic fish biomass towards the poles, both from the Southern Ocean as well as the Atlantic. So the question is, relating back to the photic barriers that I have introduced in the beginning, is this decrease in biomass, or can this be explained by a change in the light regime? And the North Atlantic is really the perfect system to study this question. It's the only deep ocean basin that is completely ice-free year-round. I've plotted Seattle here for comparison. It would be below this plot actually, and you see it is far south of where we are here in Bergen. I have spent quite a bit of time in Langbehn, where seasonality is much stronger. And for comparison, I also put Stewart Island here, which is the southern tip of New Zealand. So the perspective often is that in the Southern Ocean, New Zealand is very far south, but indeed it is not. It is at the same latitude as Seattle about. So the North Atlantic is really a unique system and can help us understand how this system will look once sea ice is retreating. Modeling is, it's pandemic friendly because it allows us to do virtual cruises at any time of the year, and that is what we have tried to do here. So we run the model along a transect that follows the continental slope here across the Norwegian Sea from about 55 degrees to 80 degrees north. And we have done or simulated along this transect two scenarios. One is the winter scenario that you see on top here, starting about a month before winter solstice and then going towards winter solstice, ending up at the northern point of the transect, and a second transect starting at summer solstice in the south and then driving north. In the first step, I'm showing again the light comfort zone based on the modeled light intensities here. So this is the zone where mesopelagic fish would feel safe, and that's where they would prefer to stay. Copperpods follow a different pattern. Copperpods in the model in winter hibernate at depth where they are dispersed, while in the summertime we see them that they concentrate in surface water where they graze and aggregate. And if we then overlay the strategies predicted by the model, then we see that for the winter scenario, they can more or less follow their light comfort zone along the entire transect. And the color of the line here gives you an indication that they're able to maintain high energy reserves throughout. There are some opportunities at the southern part of the transect to feed on overwintering copperpods at depths during daytime, not at nighttime, as we would usually expect them to do in surface waters. While in the summertime, you can see that in the southern part of the transect, they are also able to maintain high levels of energy reserves and to track the light comfort zone. And then we see this decline that we have already previously seen as the midnight sun period starts. And at the higher latitudes where we have complete midnight sun and no twilight at night anymore, it is never safe for them to venture into surface waters. So what we see is that they becoming increasingly desperate, their energy reserves drop. And what the model then predicts are this sporadic high-risk foraging bouts, leaving the safety of the light comfort zone, venturing to the surface to forage. And in the model, we also track their gut fullness at any time step. So we can really see that the smaller the dot, the less their stomachs are filled. So we can see they're getting quite desperate here, and then they go up into surface waters, fill their stomachs, go down again. But as the color here indicates in yellow, it is never enough food for them to fill up their reserves. So they're becoming more and more desperate over time. Of course, we can do the same exercise not only during 2 times over the year. We can do this basically for any combination of latitude and day of the year. And what we then see emerging here, the color again indicates the level of energy reserves. Then we see that especially at the high latitudes during summertime, this is where these mesopelagic fish are predicted to struggle to maintain a positive energy budget. I could draw the same plot with the color showing the predation rate here, because if they're getting desperate, then starvation is the consequence unless they take the risk going to the surface and feeding again. So then they rather choose taking the gamble of going to the surface water, instead staying down at depths where starvation is certain. And what we see is that this pattern matches very well with where we would expect the midnight sun period to start, so where there is no twilight at night, or where there is civil twilight, so the sun is only 6 degrees below the horizon here, so it is always quite light at night. And there is a slight shift towards later in the season with increasing latitude, which in this case is explained because there's also a shift in phenology of our zooplankton prey field. If I would take away that phenology, we would also see that there is no shift towards later in the season in the energy reserves here. The consequences of this are, because the individuals at higher latitudes struggle to maintain energy levels as high as at lower latitudes, that they will have to take higher risk. That means that the annual survival is much lower at the higher latitudes, and it drops actually very rapidly across a small latitudinal gradient when crossing the Arctic Circle here. They're also not able to maintain a high surplus energy budget here. So we can see it drops by about 2/3 when crossing the Arctic Circle again. The population level consequences are then that we predict that the Arctic waters are population things, and that there's only positive population growth at latitudes below the Arctic Circle. So this point where we change from a source population to a sink population could also be seen as the poleward range limit in this case. Then the question, what's wrong with Helsinki? Asked him. I will get back to that. Then, of course, the question is how these barriers, these photic barriers that I have just shown, will change under warming, and if they will persist or if temperature actually overrules them. To test this, I have joined forces with my colleague, with Gabriella Lundström. She has been working on herring, and she has developed a model for an apipelagic species in this case. Other than our mesopelagic species, it does not perform diel vertical migration. It is a seasonal migrating species that migrates from foraging to the spawning grounds, and then takes a huge loop through the Norwegian seas before ending up in the overwintering areas. And the models are similar in their structure and in terms of mechanisms, but the ecology of these two species is very different. And the models differ further because the herring model is resolved across different sizes, while in the mesopelagic model we consider explicitly predation mortality. So we are going to contrast the predictions from these two models. I'm going to start with the mesopelagic model because you're already familiar with the predictions. This again is what I have shown before, just more stylized form. At the low latitudes, we expect these regular diel vertical migration patterns. At the higher latitude, during the midnight sun, we expect this high risk foraging bouts into surface waters, but generally trying to stay at greater depths. With warming, the need to perform these foraging bouts becomes higher because the metabolic rate increases and therefore makes it necessary to risk foraging in surface waters more often. In consequence, we predict that these photic barriers, at least for the mesopelagic model in this case, will persist or in fact slightly shift southwards. If we look at the herring model here, just showing the large-scale foraging migration through the Norwegian Sea, this species ceases foraging at the onset of winter, then goes through a period of winter starvation before resuming feeding in spring. And the level of reserves at the end of the winter season is very crucial because it is a spring spawning stock that we have here along the Norwegian coast. So basically all the energy reserves that are left in spring is capital to fuel their spawning. Here we are comparing two different populations, one at 60 degrees and one at 70 degrees north. At 70 degrees north, the winter is much longer, so also the starvation period is longer, while at the lower latitudes, the winter are shorter and feeding can resume earlier in the season than at higher latitudes, which is also reflected in the energy budget here that is available at the end of the season. With increasing temperatures, similar to what I have shown in the mesopelagic model already, the metabolic rate increases, and therefore we see a faster depletion of the overwintering reserves. Which in this case means that to maintain the same level of energy as under the 0-degree warming, these species would have to shift south, where the nights are, or the polar night is shorter, the winter season is shorter. If we would expect this species to shift polewards, as would be predicted if we follow the thermal envelopes thinking about species tracking their climate velocities, then they would end up in the season where the winter period is even longer, and sustaining that under the higher temperatures would not be possible. Although the temperatures are lower at higher latitudes, the scaling of the winter season overrules that effect. So we see in the model that moving towards higher latitude in this case is never an optimal strategy. And what the model then predicts is that fish will have to shift southwards towards less seasonal environments, and the predictions from the model are that at higher latitudes, this shift will have to cover a larger distance than at lower latitudes, and we see the same for larger individuals than for smaller individuals. So the question is, how reliable are these predictions? We have really tried to kick the tires of both these models. So we have run extensive sensitivity analysis. For the herring model, we have tested 53 parameter combinations. The blue dots here again are the two populations that we are comparing, one at 60 degrees and one at 70 degrees north. And the red arrows will indicate the direction and magnitude of shift that we would expect under a warming scenario. And here, in all cases, for a 2-degree warming scenario, we see that they're consistently shifting southwards. There is not a single scenario where we saw a shift polewards. And across all scenarios, it is a fairly consistent shift, so we end up with a very narrow range of latitudes that we would expect them to end up. For the mesopelagic fish model, the point where we would expect this northern range barrier to lay, so this photic barrier varies with the initial parameterization. Here we have tested 73 parameter combinations. You can see there are slight differences here. It might shift a bit north, it might shift a bit south, but in generally it is still a fairly narrow range of latitudes. And again, if we apply a 2 degrees warming, there's not a single scenario where we saw a poleward shift, but in all cases, we saw that actually the direction of the shift is southwards, although many of them are rather small. So in this case, we wouldn't expect the range barrier to shift that much. I think the take-home message here is more that it will persist despite warming. So the models are both fairly consistent in these predictions that there are photic barriers, and under a 2-degree warming, we will see a southward shift. Are there questions at this point? Not yet. Good. Then I will use a few more minutes, and I promise to be brief on a last example, because not all species distributions are driven by climate. We can use this same framework also to apply it two systems where we don't yet see maybe so much of a climate-driven redistribution. And I'm going to show an example here again from the same system, from the Norwegian Sea. And I'm going to talk about the Greater Argentine. It is a species that is distributed on both sides of the Atlantic. I've just pulled here some data from the ICES database that shows the general distribution. We know this is not the full extent. I've complemented this with some data from survey reports that are not published in the ICES database, but the pattern of distribution here is what made us curious, because it seems like this species follows a very narrow depth gradient here, and our suspicion was confirmed by colleagues from the Marine Research Institute here. From the acoustic surveys, we see that this species is mostly distributed between 200 and 600 meters depth, following very closely these contour lines here. But then there are also many areas where we don't seem to find this species. So in blue, I have now just highlighted all the areas that fall in this depth range between 200 and 600 meters. And one can wonder why the species is found in some of these areas, but in very short distance, not found at the same depth intervals. And I just want to highlight 3 areas here. So Newfoundland, Iceland, and Faroe Islands. In all cases, we actually see that the leeward side, so that the current facing side is where we find the species, and the leeward side is where the species seems to be absent. So based on that, we formulated a hypothesis. So we think we can explain the distribution of Greater Argentine by looking at the topography, the light, the advection, and the prey abundances. So the idea here is that Argentine is a specialist feeding on mesopelagic scattering layers. The small mesopelagic fish that we have modeled before doing this diel vertical migrations in the open ocean, staying in the light comfort zone allows them to avoid and predation and evade even the Greater Argentine with relatively light sensitive eyes. But once advection brings these scattering layers across the continental slopes, their downward migration during daytimes becomes blocked by the shallow bottom topography, and they aggregate closer to the bottom. And this is where we think that then Greater Argentine finds a niche and the right light levels to predate successfully on the scattering layers. Then again, at shallower depths, we think that the Greater Argentine is excluded because of high predation levels from other species itself. To test this, we have just reparameterized the model that I've already shown you. Instead of the mesopelagic species, we now parameterize the model for the Greater Argentine. The mesopelagic fish enter the model now as a prey organism that we have just fixed to the light comfort zone, so they will migrate with the changing light intensities. And again, we consider a larger piscivore predator in the model. So what are the predictions from this model? This is work that one of our master students, Andy Joll, has been doing. He arrived in Norway in February and we went in lockdown in March. So he has been sitting in his student flat and has been trying to understand this relatively complex model and has done a fantastic job on this. So this is a shout out to him. This is more or less all the work that he has been doing. What we are seeing in this model is that these mesopelagic scattering layers, once they hit the shallow bottom topography, we see an up concentration of the prey densities here where they get blocked during their downwards migrations. We can overlay that with the predictions where we would predict the Argentine to migrate. There are some artifacts here that I will explain in a second, where we see that in the open water, the Argentine is predicted to go towards the surface. And we can take an in-depth look because we are tracking all the vital parameters for every time step. we can start understanding why the model predicts these species to go to surface waters here. The reason is that they never seem to be able to ingest enough prey down here. The blue ingestion is hidden by the surplus here. We see that their guts are empty. They can't build up reserves. This is because the scattering layer always evades them because they are able to stay within the light comfort zone. Then we see a range of depths where this species does rather well. We see in the turquoise line here, they are able to build up energy stores. They always maintain full reserves, and the predation rate is rather low. And then at the more shallower depths, again, we see that the predation rate is much higher here, but still they're able to accumulate surplus, but at the cost of a much higher predation rate. So we see that the cumulative survival, which is here in the lower right-hand corner on the plot, it drops drastically at the very shallow latitudes. So in this case, the models seem to confirm what we have hypothesized, that at the greatest depths, the species might be constrained because of starvation. It's simply not possible for them to survive at low prey concentrations, while at the shallower depths, predation from larger piscivorous predators could be the reason why they're excluded. How does this hold up against data again? So we can compare our model prediction with the acoustic observations. So we see a very good fit here, both from where the model predicts the species to be and where we actually see it on the acoustic surveys. But there are also some more interesting ecological questions here. The model, for example, predicts diavertical migration, which is anecdotally reported for that species, which we were, however, not able to confirm. We have been doing some dedicated trawls this year during a student cruise, and we were not able to find this species in the water column. So I think this is where we can actually make use of this modeling observation feedback loop. So we're using the data that we have gained now from the field to inform the model again. So we might need to consider to, for example, introduce a cost here, an activity cost for the migration that is not in the model, and then I'm sure that we will see no migrations here, for example. And I hope that in the future, we will be able to expand this work and also try to explain some other species distributions, such as redfish here, for example, which occurs at the same locations as Wassel, but has a very different vertical distribution in the water column. And with that, I'm coming to the end, and I'm happy to take questions if there are any.
Thank you so much, Tom. That was a wonderful presentation. We do have one question. Are you able to see that, or should I read it out?
Yes.
Once you've had a chance to take a look, if you wouldn't mind
Yes.
always rereading the question for the audience, that would be great. Thank you.
Okay. So Alex de Robertis asks, how are the latitudinal changes in population of piscivores parameterized? In the Pacific Arctic, there is a huge latitudinal decrease in predatory fish biomass. Curious if the inference related to latitudinal barrier is sensitive to this. Are there situations where low predator densities in the Arctic allow for near surface feeding behavior? That's a very good question. We have not scaled the predator density with latitude. In fact, we also keep the predator density the same across the entire water column. It just scales with the light regime in this case. I would argue if mesopelagic fish would be abundant at high latitudes, there would be also likely predators feeding on them because they are such an easy and convenient and energy-rich prey. But we haven't accounted for that specifically in the model, no. And if that would allow for near surface feeding behavior, I'm not sure. There are some observations where we know that this other type of mesopelagic fish, Marolicus mülleri, starts schooling during the midnight sun. So that might be an alternative strategy that would allow them to expand into these high latitude systems. Are there more questions? If it's possible, you might as well try to unmute. Does your energy budget model for overwintering consider the metabolic costs of sub-zero temperatures? No, it does not. But I think in our system, we don't encounter sub-zero temperatures, I think. I think for the herring model, we only model until 75 degrees north. I think in our system, at least along our transect, there are no sub-zero temperatures at those latitudes. For the mesopelagic fish, we have expanded a bit further north, and I think it's just on the edge there for around 0 degrees, but not at sub-zero temperatures, I think. Okay, then there's one more question related to the computation time for this kind of model. Example, for how long does the sensitivity analysis take? Yeah, that is a very good question. So the model is set up in Fortran already, which makes it much more computationally efficient to run. I started doing similar work on this visual light model during my master's, and my first model was coded in R at that time. And I was only running it for one smaller transect, and it took about 27 hours to run. And now with this more fledged model, it runs in a couple of seconds. However, that sensitivity analysis, because we run it in a very high resolution and for so many different latitudes, I think the mesopelagic fish model took 8-9 days to run in total. Yeah. So there is some time involved, especially this is also the reason why we only did a one at a time sensitivity analysis. If we would have tried to do more sophisticated sensitivity analysis, it would have easily taken several weeks or months to run. Yeah.
All right,
Hey,
thanks,
Tom.
Tom. Go ahead.
I had a question, which is, I guess, more of a little technical question. So how are you handling variation in water clarity in the different case studies?
So I mean, you know the Axnes and Utne model very well. So it enters through the light attenuation in the light field.
Right, but so how are you parameterizing the light? Or how are you modeling the light environment at depth? Like, are you working with in situ data sets, or are like what sort of assumptions are going into that?
So we have very limited in situ data sets. I think this is one of the caveats where we all need to get better, is to also measure other environmental factors besides temperature. I think people have just avoided light because it's cumbersome with the units and the sensors are difficult and often fail. So we have, at least for this first transect where I showed also the acoustic data, there we have some light measurements. And I have been also out in the southern part of the transect earlier this year, where we also did some light measurements, and then we do backwards calculation of the light attenuation coefficients, and these we use then in the modeling. So that's the beam, that's the diffuse attenuation coefficients.
Thank you. Great talk.
And I think what's easier for us than for your visual modeling is that because the mesopelagic fish generally stay at great depths, we only need to consider one wavelength, because light at those depths are usually monochromatic anyway. So we only focus at 486 nanometers, which makes it much more easy than visual ecology in surface waters, I think. So there's one more question for the herring model. Is your group going to incorporate the food availability before the overwintering period blooms spatial match mismatch. If we're gonna do that, I might be the wrong person to ask because that's the model of my colleague Gabriella. I'm not sure if she is actually planning on that. I know that she has been planning to develop this further in a more fully fledged life history model. We have not only focused in the winter period here, we have also run it for the entire season in this case. What we then see is that for the summer season, we would actually expect some of these range boundaries to shift polewards and in winter southwards, equatorwards. So if we consider the full season, there might be no shift at all because they basically counter each other. But so the general prediction from that model would then be that we see an increase in migration in and out of these high latitude systems. But we have not accounted for bloom spatial mismatch. No.
All right. We are right at time. So if anybody has additional questions, Tom's email is up on the slide. I know I have some questions lingering, so I'm going to follow up with you, Tom. Thank you so much for such a great talk, and thank you
You're
everyone for
very
coming.
welcome. Thank you for the invite again.